neurons that have been described for many years in the text is provide a neural correlate of work. As to say that this is not universally accepted. There is some debate. This is a screenshot from presentation of the Journal of Neuroscience that discussed in neural medicine. There are two articles. One was for expressing exactly what I described, that this persistent activity of neurons in the other areas continue to acquire action potential is what represents. Other investigators have also taken other. So therefore, in my talk today, I will just go over some of the basic mechanisms and explain why, in our view, persistent activity is, okay, this is the opposite, that persistent activity is not necessary or not sufficient. But in my talk today, I will go over this mechanism of why we think persistent activity is, in fact, the neural correlate. Over the experimental evidence that supports you and the model that sort of summarizes all of these ideas, we pump. And once we have an understanding of this basic mechanism of working memory, then I'm going to move on to the second part of my talk, which is how we can improve working memory by using brain stimulation. Let's start with a basic problem. How is it that neurons, brain, can generate action potentials even after a stimulus is no longer present and create this sort of mental? What is it? Can we design a circuit, simple electrical circuit, that would have a property like that? How difficult would that be? It turns out this is not such a difficult project at all. This is a sort of a circuit, a schematic diagram that illustrates a circuit which is based on the principle of recurrent excitation. So this diagram, each one of these triangles here represents one this neuron receives effort in the top. In the context of what I've been discussing would correspond to the response to the visual stimulus itself that excites some nerves in the retina that get propagated across the pathway of visual areas in the cerebral cortex, and eventually you arrive at that's the afferent. And each one of these neurons is also connected to the other neuron. When this first neuron is activated, second neuron, and this activity will reverberate in the. And even when we turn off this external afferent input, these neurons can maintain precision. So at least in principle, it is possible. And design a concept that will perform. And this is in fact a more realistic circuit of what we would look like basically. And again, each one of this triangle presents a single neuron, and these neurons are arranged in this circle depending on the location this neuron is. So by virtue of drawing this neuron there, to sort of indicate that this neuron is only respond to the stimulus appearing to the right side of the screen, the 0 degree. This neuron that will be activated by a at the top of the screen. It's not that these neurons are arranged like that, brain, but we're schematically representing them. Okay. So if we do a circuit like that, if I can start with it. A circuit like that. But nonetheless, okay, it's not necessary to show this image. So A circuit like that will, in fact, be activated by a stimulus appearing somewhere on the screen and continue to be active during the delay period just by virtue of the connections to the network. There is nothing else necessary. We create a network basically that maintains activity just by virtue of this. So this model provides us some additional insights. So if this model is truly how the brain things information memory. So we can use that to make some predictions. The prediction is where is the inaccuracy coming from? So we see, a stimulus like that, and and it's no longer present. We close our eyes. We don't have this crisp image of it, but we have very quickly a smear of this stimulus. That's what we try to capture with our model. Can our model explain why we lose it or others sometimes with our additional okay. So let me illustrate again the presentation of the stimulus screen, clicker, something has to remember the stimulus, and a response. So the response may not be perfectly accurate. And we know experimentally, when that's like that, you don't always make an eye movement perfectly accurate, exactly where the appears, but in fact, there may be some inaccuracies. So let's see how the model behaves and how the model changes. Now, the x-axis of the one is done. Now, the y-axis here are the different neurons. So each line here represents a single neuron change depending on the preference of the neuron. So this 90-degree rotation is this. Neurons that prefer application are going to be maximally active and the stimulus in fact appears. This activity will be maintained during this delay period by virtue of this recurrent excitation connection. However, this peak of activity, this bump of activity may drift in time. And wherever this peak ends up at the end of the day, this is exactly what the location that we call at the end is determined by where this peak goes. This is what the model did. Or in other words, sometimes in some trials, this activity, the bump activity, may drift randomly into the noise in the system to the left of the location of the stimulus. But in another trial, this activity to the right location. Okay, so this is the model prediction. If in fact this is true, this is how the brain maintains this time, then we would expect that in those trials, the subject may be responsible left, and those trials where the subject may be responsible right, in fact, have slightly different patterns of activation. Here, exactly this analysis in that we brain broke down subject in these red points that represent like two responses deviating clockwise or counterclockwise from the response of the stimulus. Then we compare the tuning functions of neurons based on the red responses, the responses. And in fact, we saw that there was a significant difference between exactly as it would be because our model in fact is what. And as you see, this prediction was not trivial. If we create a model that depends on decay rather than drifting on it, this will make very different. So I think intuitively, when we try to maintain something in memory, we have the feeling that this memory basically sort of fades away with that word basically. Then this model would in fact not predict anything at the time. So So let me go on now to the alternative mechanisms. What is called as activity silent alternative models about working memory maintenance. And not so much on this generation of persistent discharges during the period. People have speculated that maybe some changes in synaptic strength in the weight of connections between neurons may are modified when we're maintaining something in memory. That may be the mechanism. And I should say, there is some evidence. There is definitely evidence for the silent mechanism. One example of that is what is called bias. So these panels here represent again the rate response test that I described. Here somewhere, subject has to remember it, and then what happens from one trial to the next? The system that is optimal. Once you have completed one trial, best thing that it could do is completely wipe out this memory of this previous item and then focus on the next item that's going to go on. And there's no trace of this previous item. But in reality, human it's in fact an imperfection of the that is where the stimulus in the previous trial ends up influencing response to if the previous trial appeared in this X location here, the current stimulus that we're trying to remember there, oftentimes the subject will make an movement that deviates from the real stimulus or the current stimulus a little bit in that direction. Actually, this depends on the repetition of these stimuli with each other, then the response will be stimuli. These are repulsion, the previous stimulus tells the memory of the current. So in a series of experiments, we examined how the activity of neuron differs for the entire interval of 1 hour. This is shown. And the y-axis of this plot presents a measure of firing, the number of action potentials per second, normalized in square. X-axis here presents time in the time scale of displacement. So when figures delay stimulus, the activity of neuron high over here, first are tuned for this previous red lines here. The snapshot of those tuning bases. Once the sensation of point comes off of the movement is a case, and in between the two trials, the critical at some point, all this persistent activity is away, and there's no more information about the stimulus. But when the next trial begins, or the next stimulus appears, there is again the activation of this previous stimulus, which we shouldn't have a memory. This result we saw in this analysis that is not compatible with the plasma tractor model. And the best way to visualize it is that this reactivation phenomenon causes an activation in our it's a new problem of activity. When the new stimulus merges the two, and that is what causes the correction to fall in between these previous two. That was the first part of what I mentioned on the basic mechanism. Summarizing that, we pause and take some. Okay. So what I've shown is that neurons in the brain retain discharges through current. Showing that activity may drift in the system, and these drifts, time is a function of time, down in fact with behavior. Which I show you finally that activity silent mechanisms exist. They really exist. They're evident, but they influence behavior in as much as they pause here. Okay. Okay, very good. So we found ourselves in this position about 4 or 5 years ago. These latest really, and that was about the time that my lab moved to Vanderbilt, and we sat down and we thought, right? So we studied a lot about this basic mechanism. Understand how they work. Can we do something to actually improve memory? Can we improve memory for conditions that are relevant to goal conditions, but also to normal decay of function in, for example, in aging? And that's what gave rise to this idea of what we're trying to do to develop a pattern of electrical stimulation targeting some part of the brain that will allow us to improve part of the brain that we selected when it's called the nucleus basalis major. It's an area deep inside the brain at the base of the skull, and it's area inside the brain that we can target with the deep brain, and then use the non-human primate model to test if this is again a more detailed view of exactly your term. So we have a part of the brain. At the same time, we do stimulation. We also have the difficulty have some recording like this. Only the behavioral effects, also the neural effects. These slides over here are the anatomy use the reality check that are not exactly this. What we're targeting is, chemistry, so that these neurons that produce acetylcholine are encountered in the thalamus. In fact, this is our primary target. So we selected this nucleus. This nucleus contains these neurons that produce the neurotransmitter acetylcholine, and then axons from these neurons right there. Okay. Stand and predict pretty much the entire stimulating electric nucleus. We have our finger on the switch essentially, turn the acetylcholine release on and off pretty much everywhere. So determining how the stimulation is not good at all, has to trial and error, try to figure out why it's going to. I'm showing you that it works. So we use a biphasic forces so that we can have no current, current neutral delivery. So we have negative. Here's the stimulation frequency of about 80 hertz. So we have seconds. Brain are the output of disposable current years and steps last 5%. Do the modeling or follow the modeling and help to see activation is parameters stimulation, and we estimated that by applying this kind of current to activate something 1 millimeter radius. So it's a tiny part of the brain that we're activating kind of stimulation parameter. It's critical for our experiment. We don't want to spread stimulation at all, but we only want to target small signals. So now, for how long do we apply this stimulation? That was the other critical that we found here. Our first experiments just turn on this 80 hertz stimulation in a continuous fashion. So stimulation for several hours on, and had animals do these tasks. And not only that was that not effective, but in fact, this actually actively inhibited some tasks. This is all shown here. And this is so our first result was a resounding failure. We found a stimulation protocol that impairs working memory performance, and we can make these monkeys worse, basically, that they can. But then we started playing with this stimulation, figure out that if we can apply that in fashion, by something in the order of 60 or not continuously, apply for 15 to 20 seconds per minute. So 15 to 20 seconds for 60 to 80 hertz. Then we wait for 40 seconds, and then we repeat this cycle. Then, in fact, we saw we split off the baseline. This titrate the difference. So across a range of time intervals, how long these monkeys have to gain this information. I control stimulation in the black line here produced better. So we also have the ability to record neural activity from the brain, and this slide here illustrates how we did this. First, so this is a drive here. Station, stimulus station. This task we saw a second stimulus. Have 1, 2, 3, 4 of those trials. So then in the intertrial interval stimulation did not have to be sized to any specific event during task. It's obviously going to be impossible if we do that in humans down the road. We have a therapy basically that's going to improve cognitive function without having to turn it on and off precisely when you start thinking about something, when you have to. As long as we had this burst of stimulation that repeat periodically 15 seconds every minute, this burst is going to do that. And this stimulation was, in fact, let me give you one more detail about fast stimulation. So this is a more difficult. That monkeys now see 2 different stimuli. Here, have to make an eye movement. Remember, either the first stimulus or the second stimulus, depending on the fixation. Form is white, have to look at the stimulus, or the second. The fixation form, or the first stimulus, that second stimulus, or non-important task that allows us to see the effect. And what we saw then in this experiment is illustrated on the left, showing the response of a single neuron. Blue line is the control condition, orange line is the stimulation condition. What we see in general is that overall, by stimulating the cells, we saw high overall activity in the preclinical. So However, again, unexpectedly, and to a surprise, basically, what this stimulation did is not sharpen tuning functions of the neuron. So the preference of the neuron become more specific. Anything, it became broader. Activity of neurons is higher now. Stimuli that are further away from the flattened. This was perplexing for us again. We have an intervention, post-call intervention, yet we seem to be losing the tuning or the selectivity of neurons for these signals that were available. So we again interpreted that. We went back to our model, and so how would our model be? Results are shown here. So this is the all condition, the off condition. There is no stimulation. Stimulation is off. The stimulus is neurons that are active to bump off. Structure appears that we have, or some reactivation of some neurons is off. This bump of activity. And we turn the stimulation on. You see this broadening of tuning means that the bump of activity is now wider. More neurons are now activated. And by each of this broader level of model activity, more stable. Inertia. Have less density. This was precisely so experimental. Clarify them to see that this model that we have all of these really it's. However, there was a marginal case. That is a case where second stimulus closer to the stimulus close in space with each other. The model predicted that sometimes those two bones are going to move each other, then the activity will actually drift and then that we went back to the experimental result and we found out that this in fact in general it was better in working fast if they have to remember distributions that were close with each other, then the performance was higher. This marginal case, in fact, right. So other than that, then, so other than this very special case, generally we have now found parameters of stimulation are effective in improving working memory. This might be associated. They're not, sure they don't have any memory. They're young adult monkeys used in our research. Looking to see if stimulation can also be effective. Animals that are closer approximate human conditions that have problems with memory, or in this experiment, or all monkeys. Monkeys age 3 times faster than humans. 1 monkey year is equivalent to 3 monkey years. Offered for 25 to 30 years old. 75 humans basically. And we did the same thing. So we implanted these animals, stimulators. In this series of experiments, we used commercial stimulators that are used for spinal cord stimulation, and the Parkinson's stimulation also look very similar. Higher, targeted. Monkeys had to do fast You have to look at the stimulus, there's a delay period, stimuli period, the one that's the stimulus, and we can make fast drive easier with an adaptive staircase protocol. So if the mic increase in task, and if the monkey started making errors, we would visit so we can find the estimated threshold. You know, what is the delay period that the monkey could do this task with approximately 80%. So we applied then this stimulation in several, we are over 8 monkeys, and so a highly significant group effect and individual effect almost in all the animals that we so implement the performance. A critical factor for this determinant is to see how long does the effect last, right? One time using performance, does that last? This is a issue particularly involving the acetylcholine system because we know that drugs that target acetylcholine inhibitors, they're effective when you first start them. They lose all effectiveness, and then it's absolutely perfect. So we tracked over a period of 15 up to 15 months. Again, it's the equivalent of something beyond 4 human years. And the red lines here represent slide that control animals that had surgery but have stimulation. So in fact, stimulation persisted for the animal to sort of benefit throughout all this entire interval. We also wanted to see what this stimulation does at brain activation. So we obtained PET imaging. These results here are shown by it's a composite of multiple animals, and by combining left side on this unstimulated side, change in brain activity before the intervention until after the right side is the change in brain activity in the side that was stimulated. So over 1 year, generally, brain activation increased in the brain, as in fact happens with aged individuals, humans, but in the side that was stimulated, we saw a significant. Finally, we some markers show that this stimulation proves that tissue transmitter activator TPA is by biochemical marker, basically factor that you see. This is this factor is butane. Also saw that this marker is beta on ratio of to did not decrease with period of months. Generally, in aging individuals, this ratio decreases in patients with Alzheimer's or tested. So this is where we stand today, and obviously our end game here is not to improve the lives of monkeys, is to improve the lives of patients. And we, in fact, have received to try the stimulation in human patients, and we got by Medicare that they will reduce this therapy. The patient pilot study where we try deep brain stimulation targeting the patients without. It's kind of a daunting task, you know, sometimes. Sometimes I'm terrified by looking at it. So that concludes the second part of my talk. So let me then finish here by summarizing what. So I showed you that intermittent stimulation increases the body rate, unlike counterintuitive potence that and we were able to explain this problem, how that works. That causes a behavioral factor. And finally, I showed you that this is effective not only in young adults, but also in age. That this is that research and our industry partner was Constantinidis that developed us and in this experiment, and these are my clinical solutions which are solutions it's called latest in parking this.
You were previously discussing models and first blue schedule. That's now representing that more complex older model, but have there been any efforts to expand analyzing not just one potential recall event, but now these more complex patterns as with the string of numbers? I think we can memorize maybe one or two or even three digits pretty easily, but then there is some threshold you reach now where
yes, we've done these experiments. We didn't try them with the stimulation, so that's the reason I didn't show that. So the short answer to this question is that absolutely, the monkeys also have the capacity limitation just the swing set. Like famously that we have to performance goes down, and there is a limit of how many items. Context of the model that I saw, imagine that you have multiple peaks, multiple bumps being present at the same time in the model, and at some point, some of these peaks may merge with each other, or some of these peaks may, and that will explain why we cannot remember.
Mention the word reverb. This concept of I love your electrical thinking about play of reverb and sampling hold, that kind of the idea of how long you want. That was just kind of comment. Deep brain station practicalities of that, and if you think eventually we'll get to the point where Alzheimer's, you can have some that you have people that have it now, that eventually as opposed to some other modality.
Yes, so that's an excellent question. So the answer is yes. I mean, with the present state of technology, the deep brain stimulation, is our best bet, our best approach. The therapy is going to be very similar to the therapy that patients are having movement disorders. For Parkinson's disorder, it is now standard of care to have deep brain stimulation. We have a stimulator it is invasive, but 20 years of Parkinson's disease development basically has really solved a lot of the problems. Certainly thought about ultrasound or something non-invasive that doesn't require as often. Problem with that is, first of all, to see the effectiveness of the therapy, nothing rivals the precision of Kevin like that. But down the road, that would mean that these patients will have some kind of helmet that they is maybe practical. However, once we have that and we found that this is working exactly where we need to target. It may be possible that this patient wear the helmet for one of the basic therapy and take off. So entirely possible that that.
Like 10-minute therapy a day or something.
Yes, yes.
You're, something physiologically like a muscle, right? Have all these things, all these neurons available, right? Process. And is that your feeling of down at the point of that application?
That's right. And I had the same question earlier this morning about that. Why is it that, so in aging, in Alzheimer's, or whatever it is, walls of connections to the basal forebrain, and we have some also neurons, we have some atrophy, less activation that is needed when that is optimal. We are compensating this by activating brain neurons and by activating more neurons.
Parameters to the spinal cord. FDC Boston Scientific is
For 601 students, just know that your test reports have been sent back